Dr. Stanley Chi's CRISPR lab set out to make the CRISPR kits with one clear mission in mind: to make CRISPR available to all. As one of bioengineering's most powerful tools, CRISPR is already making headlines in agriculture, medicine, and research. But in order to realize CRISPR's full potential, students and educators alike need to experience the material firsthand. That's where our CRISPR kit comes in. Principally, CRISPR only requires three things: the target DNA, the gene that we want to edit, the guide RNA, or the gRNA for short, which is roughly 20 nitrogenous bases long that is complementary to the target DNA. Like its name suggests, it is the guide RNA, so it serves as the GPS for locating the gene that we want to edit. And lastly, the Cas9, which is a protein that serves as the molecular scissors for cutting out the gene that we no longer want. CRISPRkit uses CRISPR-I, which means CRISPR interference. This type of CRISPR uses dCas9, or dead Cas9, meaning that when we go to remove the gene, we just turn it off with a molecular off switch rather than removing the gene altogether. This means that everything we do within CRISPRkit is reversible, making it much more safe compared to traditional gene editing. Today we will be running the Melanin kit. However, it is important to note that there is also a bioluminescence kit, single color chromophore kits, and multicolored chromophore kits. Before we begin the experiment, it's important to remember our labeling. We need to label a total of 4 tubes. The first two should be NC and PC for negative control and positive control respectively. Then NG for no gRNA, and then GE for gene editing, or simply G for gRNA. One key element of the Melanin kit is the CFS, or the cell-free solution. If you're a student, you shouldn't have to worry about mixing it, as your teacher should have already done it. You're going to want to add 5 microliters to the negative control tube and 8.8 microliters to each of the 3 other tubes. Next, you're going to want to add tyrosine. You should be able to find this substance in your tube labeled A. You're going to want to add 2 microliters of this substance to the no guide, guide, and positive control tubes. Next, we want 2 microliters of copper pipetted into the very same 3 tubes. Now we're going to want to add 2 microliters of the tyrosinase to each of the same 3 tubes that we just worked with. This, paired with tyrosine, is an example of an enzyme-substrate reaction and will form the melanin that we want to edit later on. After putting in the tyrosinase, we're going to be done with the positive control tube for a couple moments. From here, we're going to want to put 2 microliters of dCas9 into the guide and no guide tubes. This is what allows our CRISPR mechanism to do the gene editing on the melanin. In tube E, we'll find the gRNA, which will be added to the GE tube. This will show the dCas9 what the target DNA is and allow the dCas9 to do the gene editing. Without the gRNA, the dCas9 can't do its job, which will explain the visual difference between the GE tube and the NG tube. We're almost done with the Melanin CRISPRkit experiment. All that's left to do is to add water and wait for the reaction to occur. We want to add 20 microliters of water to the negative control tube, 10.2 microliters of water to the positive control tube, 8.2 microliters of water to the NG tube, and 6.2 microliters of water to the GE tube. That's it for the student protocol of the CRISPRkit Melanin Experiment. All you have to do now is wait 24 to 36 hours and wait for the reaction to complete. Long story short, In the negative control tube, we should see no clear difference in change of color because there is no enzyme-substrate reaction forming melanin. However, in the positive control tube and in the no guide tube, we should see a blackish, dark brownish looking color because there is that reaction that forms the melanin, but there isn't the full CRISPR setup that removes that gene from expression. However, when we look at the GE or guide tube, we will see that change in color back to the clear liquid because it does have that dCas9 and the gRNA. Additionally, you can do further data analysis by using the CRISPRkit app. All you have to do is take a photo of the 4 tubes and it will return real, concrete, quantitative data to you in the blink of an eye. All that's required to do is to label each of the 4 tubes correspondingly with NC, PC, NG, and GE, or G, as seen here. Let's say you're not using the Melanin CRISPR kit, but a different CRISPR kit. What will the results look like? If you're using a single color chromophore kit, it'll look like whatever solid color you're using, and then the GE tube will be clear. However, if you're using the multicolor chromophore kit, the NG tube will look like a combination of whichever 2 colors you're using, and the G tube will just be one solid color, as the gRNA and dCas9 will delete one of those colors from being expressed. The same principle applies to the bioluminescence kit. The NG tube will be bioluminescent and glow in the dark, but the GE tube or the G tube will not be bioluminescent and be a clear liquid that does not glow in the dark. One common question is if whether or not students are required to wear gloves when working on the CRISPR kit. Although it is not required because none of the reagents are toxic or dangerous to the touch, we do recommend it because it is good lab practice. Another commonly asked question is how many students should be in one group. Our answer is 2 to 4 groups, as the reagents are designed so that 3 groups will share them at one time. Another commonly asked question is how the students can get all of the liquid at the bottom of the tubes. If the school has access to a microcentrifuge, we recommend spinning down the tubes before the experiment is concluded. However, if the school does not have access to microcentrifuges, we recommend flicking the tubes with the fingers until all visible liquid is down at the bottom of the tube. Additionally, if you have any further question about protocol or conceptually, you can consult our CRISPR chatbot on the CRISPRkit app, as shown here. Our main goal in the creation of the CRISPR kit was to democratize the technology that will change our lives for the decades to come. By spreading access to this technology, we hope to inspire the next generation of scientists that will find novel solutions to the world's most pressing problems.